when you've got sodium channels that open in the membrane, it allows sodium ions to flow in. That causes the negative nerve charge, which is supposed to be about negative 70 millivolts, to become positive. At the end of the action potential, you've got repolarization of the cell where potassium leaves to get it back to negative. So here we've got the action potential moving down the axon from left to right. Action potential moving down from left to right. We also have the propagation of the follow-up, the repolarization of that cell through the through the potassium channels opening to reestablish that negative. So why is it that a signal only travels in one direction? The answer to that question is we have a gradient that's established, a voltage gradient, and as that voltage gradient changes, the ion concentration gradients change. And you are not able to send a new signal down that axon until the sodium-potassium pump has reestablished the ion gradient back to where it's supposed to be. Originally, there's lots of sodium outside the cell, lots of potassium inside the cell. As the action potential travels down, you end up flip-flopping those. And the sodium-potassium pump is what enables you to reestablish the concentration gradient of sodium outside, potassium inside. I believe that the animation will make this more clear. Questions? I can see Reagan's face. It looks like you have a question.
Well, you said it's going to be clear with the animation.
I hope the animation will help. Yeah, just stay with me, and if it doesn't help, then we'll move on. Or I'll then at that point answer that question. All right, so the last part we need to know is synaptic transmission. And there are 8 things you need to be able to discuss with this, but there are 6 steps. There are 6 steps. And there are 8 pieces of information you need to be able to discuss in an essay question about this. So let's talk about the synaptic gap first. We know that gaps exist between axon terminals and dendrites of the next cell. These are referred to as presynaptic neurons and postsynaptic neurons. The presynaptic neuron is the signal that has already propagated down the axon and has reached the axon terminal. The postsynaptic neuron is the one that appears after the synaptic gap. And it's the one that's going to receive the signal at its dendrites. So, looking at this, are you recording this, by the way, Jackie?
Yes.
All right, that's cool. So, send that file to me when you can.
Okay.
Okay. So, looking at this in more detail, if we have an axon terminal here from this cell, and we have dendrites from the postsynaptic cell, we zoom that image in. This is the axon terminal here, right? The end of this nerve cell. This is the beginning of the next nerve cell, so this is the dendrite. And we have a gap in between here called the synaptic space, the synaptic gap, also referred to as the synaptic cleft. So there are 6 steps in the process of passing a signal from one cell to the next. The first step is that the action potential reaches the axon terminal. So we know that action potentials travel down the axon, and when it reaches the axon terminal, that's step 1. The action potential reaches the axon terminal. Step 2 isn't diagrammed on here, so I'm going to add it. Step 2, calcium channels open and allow calcium with a 2 positive charge to enter the cell. So when the action potential reaches the axon terminal, it stimulates these voltage-gated ion channels. What does that mean to be voltage-gated? Well, it's an ion channel. It's a protein in the membrane that's an integral protein spanning the membrane, serves as a channel for an ion, calcium ions in this case. Ions are charged particles, charged atoms. So we've got calcium ions that are going to travel through this channel protein. It is voltage-gated, meaning that when the action potential reaches this point in the neuron, because of the voltage change as a result of the action potential, we allow the calcium ions to enter the cell. So that's step 2. Calcium ions pass through voltage-gated ion channels. Step 2.
Can you say that one more time?
Yeah, I'll go through both of them. Step 1, the action potential that has traveled down the axon and the presynaptic neuron reaches the axon terminal. That one makes sense? Step 2, voltage-gated ion channels that are activated by the action potential allow calcium ions to enter the nerve cell. And again, we're speaking about the presynaptic neuron here. That's step 2. Step 3, vesicles which contain neurotransmitters undergo the process of exocytosis. So as a result of calcium ions entering the cell, the vesicles undergo exocytosis. If you're interested in how that process works, study option E, neurobiology and behavior, in the spring, and you can dig really deep with your choice of options. Choose that option. You'll learn more about how calcium affects this. So step 3, exocytosis. Step 4, diffusion. When we release neurotransmitters from the presynaptic neuron into the synaptic gap, there is a high concentration in the gap now and a low concentration in the postsynaptic neuron. So the neurotransmitter diffuses across the synaptic gap from the presynaptic neuron to the postsynaptic neuron. Questions about the steps so far? How many do we have? How many are we going to learn? 6. Okay. And they'll award you 8 points for this question if you can discuss some of the information that I'm giving to you in your explanation. So if you list the steps and you say what it means in each step and how it works and you use the terminology appropriately, you'll earn extra points. Questions so far? No? All right, so step number 5. After diffusion of the neurotransmitters, they bind to receptors on the membrane of the postsynaptic neuron. So the neurotransmitters that have diffused across the synaptic gap bind to receptors on the postsynaptic neuron. This is in the dendrite of the postsynaptic neuron. When these neurotransmitter molecules bind, they can either excite the next neuron and cause it to form an action potential, which would then proceed down that next neuron in the sequence, or alternatively, they can inhibit the next neuron. And here's a couple of examples. And if you state these examples, you'll earn those extra points on this essay question. Acetylcholine is an excitatory neurotransmitter. These little orange balls in this diagram, when they're released and they travel from the presynaptic neuron across the synaptic gap to the postsynaptic dendrite, they stimulate the membrane to open up sodium channels, which we know is the first step in the action potential, allowing sodium to flow into that new nerve cell and generating a positive voltage inside the cell. There are inhibitory neurotransmitters. One example of this would be, so let me write up acetylcholine as an excitatory. GABA is an abbreviation for a long term that we can talk about when we study this in more detail later. But GABA is an inhibitory neurotransmitter, and it prevents the next nerve in the sequence from firing that action potential down the axon. The way that it does it is, instead of making the next nerve permeable to sodium, it makes it permeable to chlorine. What charge do chlorine ions have?
A minus.
A minus charge. So you're going to take that membrane potential at -70 and actually lower it towards -90. Now remember that action potential we talked about yesterday? -70 millivolts is the resting. What's the undershoot? -90. So if you get down to -90, it's not going to be able to fire down the nerve cell. So both of them make the postsynaptic nerve permeable to ions. But the type of ion it's permeable to either excites the next neuron or inhibits the next neuron.
Excites is just the process.
It causes the action potential to form in the next neuron. That's what excites me. Yeah? Okay. All right, so I know this isn't nearly enough, and I've gone through with lots of verbal language and not a lot of visual stuff, so I'm going to show you 4 animations, and we'll pause them throughout and talk about it. What's up?
What's the first one? The acetylcholine, is it an inhibitor?
It's an excitatory neurotransmitter. So it actually causes an action potential in the next cell. This one causes an action potential in the next cell. This one inhibits an action potential in the next cell.
Wait, what is step 6?
Let me go through them in order. Step 1, action potential reaches the axon terminal. Step 2, voltage-gated ion channels open and calcium floods into the nerve cell. Step 3, exocytosis of neurotransmitters. Step 4, diffusion of neurotransmitters. Step 5, the receiving of neurotransmitters by the dendrite. Step 6, you've got the actual mode of action that occurs. So either a new action potential is generated in the next nerve cell, or you have an inhibitory response. Okay? Questions before I show you the animations? Nope? Okay, so again, these are the objectives. We're going to be done with them at the end of this week, making sure we understand all 6 of these. Let me show you the animations real quickly. So the first animation I'm going to show you is about nerve impulses and how they travel down an axon. So we're going back to the idea of an action potential. This is going to be an animation showing you sequentially what goes on in a nerve cell that allows the signal to move from the dendrite to the axon terminal.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location.
All right, so let me talk to you about what that stimulus can be. It can either be, they showed just a yellow kind of circle, and they caused a disturbance in the membrane. That can be sensory input from your environment. So these could be sensory nerves, and when you touch the table, you've got a change in the membrane, a disturbance in that membrane, and it allows the sodium channels to open, and the nerve to pass the impulse to say, I'm actually touching something right now. Or it can also be from another nerve cell. And what we just learned about is that what would cause the sodium channel to open and the postsynaptic neuron would be neurotransmitters from the previous nerve cell. So those are 2 ways to initiate this process. Yes? Questions so far about the video?
If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential, and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
All right, what questions do you guys have about the propagation of a nerve impulse down an axon? Yes, PJ.
How does it get the potassium back in afterwards and
Yeah.
not bring the charge back up?
So it brings the potassium back in afterwards via the sodium potassium pump. The sodium potassium pump removes 3 sodium molecules from the inside and moves them to the outside, and simultaneously brings in, so it changes its conformation. And I'm going to show you an image of that, where it pumps out 3 sodium ions and brings in 2 potassium ions.
Oh, okay.
And that's actually one of the other videos I'm going to show you. Yeah.
Where did they get the sodium and potassium from? Is it just floating around?
Yeah, that's a good question. In these videos, it looks like it's floating around, right? But we know that it actually comes from, initially, dietary substances that are broken down. But someone asked me earlier, what's surrounding nerve cells? Really depends on where the nerve cell is located. If the neuron's in the brain, if it's a sensory neuron, if it's a motor neuron, surrounded by extracellular fluid, and in that fluid, you've got these ions. So the next one I want to show you is to talk a bit about an action potential in more detail. So that first one was propagation down an axon. This one's a very similar video, but I think it has some merit in that it shows it in a different diagram.
An action potential, depicted as a red band, is propagated in one direction along the axon. During an action potential, the inside of the cell membrane becomes positive with respect to the outside. An action potential generates local currents that tend to depolarize the membrane immediately adjacent to the action potential. When depolarization caused by the local currents reaches threshold, a new action potential is produced adjacent to the original one. Action potential propagation occurs in one direction because the recently depolarized area of the membrane is in absolute refractory period and cannot generate an action potential.
Okay, so the idea in this diagram is that the membrane proteins that are adjacent to the action potential are voltage gated, means they're affected by the charge. So when you bring in a lot of positive, it opens up gates that are further down the membrane, causing that action potential to proceed down the membrane. Yes?
So the little positive and negatives are calcium, or not?
Sodium, yeah, sodium coming in would make it positive, potassium going out would return it back to negative.
They're not just little pluses.
Right, no, they're not little pluses. Yeah,
So it's kind of like a momentum thing, in that as it goes, it opens further so that it can keep going.
yeah. And that keeps it moving in one direction. It can't go back the other way because the ion gradients are backwards in that case. So instead of having a high concentration of sodium outside and a high concentration of potassium inside, after it moves through that region of the axon, they're flipped, and the sodium potassium pump reestablishes those gradients. Yeah.
So then, once it reaches the axon terminal and the neurotransmitters go into the receptors, is that when it starts saying open up the next?
Yes, that's when it initiates. So if you're looking at this as one nerve cell, if this is our postsynaptic neuron and we've got a presynaptic neuron, when the neurotransmitters hit the dendrites and it causes, if it's an activation sequence, if it's a molecule like acetylcholine that's an excitatory neurotransmitter, it's going to cause this region of the cell to be more permeable to sodium, which is going to change that membrane potential from negative to positive and initiate this process. Does that answer your question?
Yes, thank you.
All right, 2 more videos, and then we're moving on to Group 4 project. The next video I want to show you is specifically about the sodium potassium pump. So this happens after you've got an active potential. Your ions are in the wrong place. They're supposed to be sodium outside and potassium inside, but the action potential flips that so that it's actually backwards. And you have to reestablish these concentration gradients. The sodium potassium pump, which uses ATP, is a way to do this.
The sodium-potassium exchange pump is an example of an active transport process. Active transport processes can move substances against a concentration gradient, and they require energy expenditure in the form of the breakdown of ATP.
The sodium-potassium exchange pump moves sodium ions out of the cell and potassium ions into the cell. The carrier protein for sodium-potassium exchange is embedded in the cell membrane. 3 sodium ions and an ATP molecule can bind to the carrier protein on the inside of the cell membrane. The ATP is broken down to ADP and phosphate. Simultaneously, the carrier protein changes shape and the sodium ions are transported across the membrane and released on the outside. In its new conformation, the carrier protein can now bind potassium ions on the outside of the cell. 2 potassium ions move into the carrier protein from the outside of the cell membrane, and the phosphate attached to the carrier protein is released. The carrier protein changes shape again, reverting to its original conformation and delivering the potassium ions across the membrane to the inside of the cell. It is now ready to repeat the process of sodium-potassium exchange.
Okay, so this reestablishes that concentration gradient of sodium outside the nerve cell and potassium inside the nerve cell. Question?
So it's the phosphate that activates
Yeah.
that, but then how, like, if it goes sequentially, how does the phosphate or the ATP know to go?
So how does the ATP know to hit that membrane protein? I don't know. It's a good question. That's the one I don't know. Go ahead.
So maybe it's just me, but it seems like this would take a while. How long is it between these
Yeah,
action potentials?
it's a good question. So you know that action potentials take about 4 milliseconds to produce. You know that there's tons of these. They're showing you one here, right? But you got to picture millions of pumps in the membrane. So although one pump may take a while to reestablish that gradient, if you have a lot of them working together, you can actually reestablish the gradients very quickly. An exact time, I don't know. But I can tell you it's fast. Because what happens is, frequency is what determines the strength of a nerve signal. When you get an action potential, that's the maximum value. It's never going to get higher amplitude. It's only going to give that signal. It's about how frequently that signal comes to the nerve cell that determines the strength of it. Does that make sense? Right? Yeah, it's not like a slinky that you can like go higher and higher amplitude. This has one amplitude that passes down the axon. Okay? So it's how frequently you're going to make that jump. So it does happen very, very quickly. Yeah?
So does the phosphate just sort of function, I know it's not inhibiting, but like a non-competitive inhibitor where it just changes shape enough for the other stuff? Is that what its role is?
So the role of ATP is when you break the bond between the phosphate groups, you actually have electrons available for bonding, which when those electrons then bond to the protein molecule,
So it's
they
not-
change the shape of the protein.
So it's not the phosphate that's bonded to the, it's just the other electrons?
Right. What other questions do we have? Julie, I think you had your hand up. Pretty good. Okay, one more video and we're done. Last video is going to show you the synaptic transmission from one cell to the next. Alright, so the yellow in this diagram represents the presynaptic neuron, the axon. So that's the end of the axon of one nerve cell. The big blue that dips in here represents the dendrite of the next cell. So how does a signal pass from one nerve cell to the next nerve cell? That's what this is about.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
Alright, so I'm going to show this one again because it goes really fast. I wanted to explain the steps in order before this so you could see it. The one thing they're speaking about, it's a little, the word may be confusing, is ligand-gated. We've learned about voltage-gated. So when there's a change in that voltage from negative to positive, it causes more and more membrane proteins to open up. Ligand is just a chemical signal. In this case, the chemical signal is acetylcholine. So when that chemical signal is received, that ligand binds to the ligand-gated channel, and it opens up the gates. And that's what it means by ligand-gated. We'll show this one more time, take questions, and we'll do group 4.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Acetylcholine molecules diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium ion channels. This causes the ligand-gated sodium ion channels to open, and sodium ions diffuse into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
All right, so if there's enough sodium flowing into the new cell here, the next one in the sequence, it becomes positive and then initiates that action potential to travel down the axon. All right, 6 things you got to know about nerves. If you want to know a whole lot more, study option E. If you're like, I hate nerves, I don't want to learn any more, just know these 6 things at a minimum. Questions? If you can explain this last one right here, that's an 8-point essay question. But they just went through real quickly. Yeah?
So is the acetylcholine just already in the vesicles? So why is that?
Yeah, so the neurotransmitter is already in vesicles. There are enzymes that form the neurotransmitters, and then they are packaged in vesicles. I believe, I need to look it up, I think it's by the Golgi apparatus. Typically that's the role of the Golgi, but I would need to check that. So was there a question there?
So are there different, because you said there's that other...
Yeah, inhibitory neurotransmitters, GABA?
So which ones, is there like specific...
Yeah, there are pathways for which one gets released by a nerve cell. And that's a bit more complicated than we need to know for now. Again, if you want to study option E, you can learn a lot more about it. Or you take neurobiology in college. But for now, there's different pathways. What else?
Where can we find this video?
I'm going to post it on the website. I've been trying to work on updating your nerve page. All the items here on the left are different animations, and at the bottom of the animations, I'll scroll down for you. Sauerkraut, I think you're going to like this one. Every one of these on the left-hand side of these animations, when you click open the animations, at the bottom there are practice questions. Based on the animation, you can submit your answers and they'll tell you whether you're right or wrong. So Friday, I'm going to give you actual IB practice questions. There's going to be 10 of them to check your understanding of nerve cells. That'll be on Friday. And then also on Friday, I want to talk a bit about muscles. So we're going to move away from nerves, finish that up Friday, and move into muscles. All right? I would ask at this point you get in your group 4 project groups.